Me neither, I can say to you. Is it on? Yeah? Okay. Good afternoon. Today I have the pleasure to introduce you to Dr. Peter GASKILL from the Department of Pharmacology and Physiology of Drexel University College of Medicine. And just briefly to tell you that Dr. GASKILL is an assistant professor, and in his very brilliant career, we can say, he received numerous awards and honors. And I would like to mention just a few of them. Drexel University Career Development Grant, for example, top citated article in Cellular Immunology, Mentor Research Scientist Career Development Award, etc. He published numerous papers in various very important international journals, such as PLOS, Journal of Neuroinflammation, Cellular Immunology, Journal of Neuroimmune Pharmacology, etc. And today I will just tell you 2 or 3 sentences about the work that is currently done in his laboratory. So basically the research activities in the laboratory of Dr. GASKILL are focused on adaptations in the development of HIV neuropathologies. And they are examining the changes in the HIV infection processes in macrophages. The macrophages are actually the cells that are very important in the CNS because they are the primary target of HIV. And their response to this infection, to HIV infection, is actually the central component of development of various HIV-associated neuropathogenesis. And further, let's say, in addition, this group examines also the impact of drug abuse on the development of HIV-associated neurological disorders, and they are prevalently focused on dopamine, because so far they have confirmed that elevated levels of dopamine have the ability to increase let's say, susceptibility of macrophages to HIV infection. But more of that we will hear today in this seminar that is entitled Role of Dopamine in HIV Infection of Central Nervous System. So, please.
Thank you so much for that wonderful introduction. She told you a lot of what I'm going to talk about, so I don't know if I even need to talk. You could come up here, you could finish it for me. It'd be just fine. So if I speak too fast, please tell me, because I speak very quickly. And all of you, your English is far better than my Italian. I can't say anything in Italian, so I really appreciate you coming to listen to me speak about this in English. So if there's anything you don't understand, please raise your hands, stop me, and I will try to explain it again. Okay? Okay. All right. So, as Natasha said, I'm going to talk to you today about the role of dopamine in HIV infection in macrophages. First, I'm going to give you a little bit of background. I don't know how much people in this room know about HIV infection of the central nervous system, so I'm going to give you a couple of slides worth of background to bring us all to the same page, and then I'm going to get into the research. So, around 37 million people in the world have HIV. And many people don't know, but HIV enters the central nervous system within about 3 days of initial infection. And more than 90% of people infected with HIV have infection in the central nervous system. It's probably more like 100%, but we don't look at everybody. So in the central nervous system, macrophages and monocytes, so myeloid cells, also microglia, are the main targets for HIV infection. Astrocytes can also be infected, but to a lesser degree. So most of the viral production and the production of inflammatory factors is due to the infection and the response of macrophages and monocytes. Monocytes are thought to carry HIV into the CNS when they transmigrate across the blood-brain barrier, where they mature into macrophages. And so this is a way the virus gets into the brain as well. So one of the most important factors about macrophages and monocytes is that they contribute to the development of neuroinflammation due to dysregulation of cytokine and chemokine production. And so you have the elaboration of inflammatory factors from macrophages and monocytes that contributes to the development of neuroinflammation, which ultimately leads to the development of HIV-associated neurological disorders, which we also call HAND. In addition, I'm not going to talk about it today, but something that's kind of a hot topic in the field is that macrophages can act as reservoirs for HIV in the CNS. When you treat HIV with antiretroviral drugs, we are fairly good at eliminating HIV in the periphery, but in reservoirs, for instance, in the central nervous system or in lymphoid tissues, it's very hard to get at the HIV that is inside the DNA of long-lived cells like macrophages or memory T cells. And so the idea that HIV can be stored in the central nervous system in the macrophages as a reservoir is something that a lot of people are studying right now. Right? Importantly for this talk and also for the understanding of HIV infection of the central nervous system, neurons are not infected with HIV. They can be damaged indirectly due to the neuroinflammatory factors that are released by infected macrophages, so elevated levels of cytokines and chemokines, but also elevated levels of viral proteins. CNS infection leads to hand, HIV-associated neurological disorders in between 40 and 70% of infected individuals, depending on the study that you look at. And importantly, the prevalence of hand is increasing despite antiretroviral therapy. What that means is because antiretroviral therapy has been so successful, people who are infected with HIV are living much longer, and so many more people have HIV-associated neurological disorders, even though the number of people being infected is slowing down. So, as Natasha mentioned, we also examine drug abuse, and the reason we do this is drug abuse has been and is still an important cofactor in the HIV pandemic. Drug abuse correlates with risky behaviors, which often lead to infection with HIV. Drugs abuse may accelerate HIV disease progression. Whether or not they accelerate the progression of HIV-associated neurological diseases also depends on the study you look at. Most people think they do. Some studies find that they don't. In addition, drug abuse and HIV infection do contribute to neuroinflammation, which is thought to contribute to hand. The reason that I study dopamine is that all drugs, different types of drugs, opiates, psychostimulants, all of them, exert their addictive and reinforcing effects in the elevation of CNS dopamine. So, as you probably all know, dopamine is a major catecholamine neurotransmitter in the CNS, and the actions of dopamine are mostly mediated by dopamine receptors. There are 5 subtypes of dopamine receptors: D1-like receptors and D2-like receptors. And the subtypes have mostly distinct pharmacology, although I don't know that that is so much true anymore. The classical pharmacology of dopamine receptors, the D1-like and the D2-like, are very distinct. As we'll talk about later, there's a number of alternative pathways that are now being examined, and those can be similar between the two types. The concentration of dopamine in the CNS and the effects of the dopamine on the dopamine receptors are regulated by the dopamine transporter and other dopaminergic proteins, which control the amount of dopamine that the receptors have access to. So, dopamine and HIV-associated neurological disorders have been entwined and examined together for the length of the epidemic. So, in patients who are not on antiretroviral therapy, there are high viral loads, which means a lot of virus, and increased neuropathology in dopaminergic regions of the CNS, such as the striatum, other parts of the basal ganglia. There's substantial degeneration in the substantia nigra and the basal ganglia in HIV-infected individuals. And there's an increased dopamine. If you look at the macaque model of HIV, so for the models of HIV, we don't use rodent models because rodents cannot be infected with HIV. So generally you use macaques. If you look at the macaque model of HIV, you can see increased dopamine metabolites in the CSF of macaques. And if you treat macaques with drugs like selegiline or L-Dopa, which increase the amount of dopamine in the brain, you see an accelerated neuropathology in the brains of these animals. In the CART era, which means since we have started to give combinatorial antiretroviral therapy, which is about in 1996, we find there are still significant changes to the dopaminergic regions, but the changes are more subtle and require different technologies to observe them. We've also seen that CNS dopamine is increased in HIV-infected individuals. Actually, this is somewhat controversial. A lot of studies show there's a decrease in dopamine in infected individuals, but most of those studies were done on post-mortem individuals. If you look at studies that look at the CNS of alive HIV-infected individuals, the CSF shows higher levels of dopamine, and some people think it's associated with particular mutations to the DAT for the dopamine transporter, the 1010 genotype, which is associated with risky behaviors, has also been associated with the increased CSF dopamine in HIV-positive individuals. So a little bit more specifically on dopamine and HIV and macrophages and monocytes. So macrophages and monocytes do express the dopamine receptors and all of the other dopaminergic proteins. Dopamine modulates HIV infection and function in macrophages in a number of ways. So it increases HIV replication and increases HIV entry into the cells. It can also modulate signaling and cytokine production, which is research that I'm not going to talk about today really. And the activation of dopamine receptors significantly increases monocyte movement and transmigration within the CNS. And this was done by some colleagues of mine. I'm also not going to talk about that today. Notably, many drugs of abuse have also been found to increase HIV infection in macrophages. So if you have macrophages and you treat them with cocaine or methamphetamine or heroin and look at the level of HIV infection, you will also see an increase. But this is not the same mechanism that I am talking about, because those drugs which mediate increases in dopamine when they're given in vivo do not do so in monoculture of macrophages. There's only macrophages in the dish. It's unclear what effect cocaine is going to have. So I'm not studying what is going to happen with the drugs directly to the cells. I'm studying dopamine because that is the outcome of taking drugs when you are alive and you are infected with HIV. All right. Was the background clear? Was there anything else, anything that anyone wants to ask about? Okay. All right, so before we get into the research, I just want to go over the experimental methods my lab uses so it's understandable for everyone what I'm talking about. So the way that we do this work is we isolate PBMC, or peripheral blood mononuclear cells, using phycol isolation from leukopaks. So we get blood from the New York Blood Center. We isolate the peripheral blood mononuclear cells from the blood. Then once we've isolated them, we culture them for 6 days on tissue culture dishes in the presence of macrophage colony stimulating factor. This allows the cells to mature into macrophages. And then we do one of two things. Over here on the right is when we want to just characterize the cells, we'll isolate the cells and then either lyse them, collect the RNA, collect the protein, treat them with dopamine and do these things. And that allows us to characterize them by qPCR or ELISA or FACS or Western blotting. If we want to examine infection, we will plate the cells in different multi-well plates and either do replication assays where we infect them with HIV and then collect the supernatant every day for a certain number of days afterwards and measure the amount of viral proteins produced, and that is a surrogate for replication. Or we will run what we call an entry assay, which is hard to see with the light, but basically you infect the cells with HIV and the cells turn a different color depending on whether or not they're infected, and you can measure infection quickly over the course of 1 day. And I'll show you both of these experiments over the course of the talk. All right, so a few more details that I want to talk about before we get into the research. It's important to note that as anyone who works with primary cells knows, there's a lot of variability. When you work with primary cells, they're derived from different people. So if you work with cell lines, often you can do experiments, N of 3, you're done, you have beautiful error bars, they're this big. If you work with primary cells, this does not happen. You need much larger N's because the variability between people is quite large. So what I'm showing you here on the left is infection of macrophages from 3 different individuals with HIV. One of the things you'll note is if you look at the y-axis, you might not be able to see it, but the y-axis on top, the highest value is 1,000. The y-axis in the middle, the highest value is 2,500. The y-axis on the bottom, the highest value is 8,000. The slope of the curve is about the same. The infection, type of infection, the phenotype is about the same, but the magnitude of the infection differs with the cells. I'm using the exact same virus here, the exact same protocol. They were all infected at the same time in the same dish. So what I'm showing you here is different people have a different susceptibility to HIV infection. In addition, different people have different sensitivity to dopamine, so many of the infections we do, we use multiple concentrations of dopamine because we're never sure how much dopamine the virus will be exposed to in the body. And it's also important to note that while some studies show that dopamine is cytotoxic, in our hands it is not cytotoxic, and we've done experiments out to 24 hours, and macrophages don't die in the presence of dopamine. All right. So now we're going to get on to the meat of the talk. So the talk is structured as a series of questions that I pose and then I attempt to answer. Right? So the first question is, what are the effects of dopamine on HIV replication in macrophages? You already know the answer to this because I already told you. But basically, the answer is that dopamine increases HIV replication in primary human macrophages. So let me orient you to this graph a little bit. So what you're looking at on the left axis, so the y-axis, is the concentration of P24 in picograms per mil. P24 is a protein that makes up the viral capsid. You can measure the amount of P24 released by cells in the supernatant as a surrogate measure for replication. So the more the virus replicates, the more P24 will be in the supernatant, right? On the x-axis is the days post-infection. In this particular experiment, we collected every 2 days out to day 13, right? Throughout the talk, all of the infections that are controls, cells that were infected with HIV alone, no dopamine, are in red. In the rest of the talk, all the infections that were done in the presence of dopamine are in different shades of gray, depending on the concentration of dopamine. So what you see here, the gray bar, is an infection of macrophages from a single donor with HIV-YU2 in the presence of dopamine. The red bar is the infection in the presence of just HIV. What the graph is showing is that in the presence of HIV plus dopamine, there's a significant increase in the amount of replication that we see out to day 13. One of the notable things about this is here on day 0, we add the HIV and dopamine, and on day 1, the next arrow, we wash it off. So for most of this graph, there's no dopamine in the culture. The effect of dopamine is happening due to exposure to dopamine for 24 hours. And really, if you know anything about dopamine, the exposure to dopamine is probably an hour or 2. The dopamine is breaking down in the media and forming metabolites, right? So somehow, the presence of dopamine for an hour at the beginning of this process is causing this effect that goes out 13 days. To note, I don't know how much you guys know about HIV, but just as a side note, HIV is generally classified in 2 different ways depending on the co-receptor that it uses. When HIV infects a cell, it infects using the CD4 and then either CCR5 or CXCR4. So viruses that use CCR5 are called R5 viruses, and viruses that use CXCR4 are called X4 viruses. In general, viruses that infect macrophages are R5 viruses, and all of the viruses I'm using in this talk are R5 viruses. So YU2, for instance, is a virus that was derived from brain tissue of someone who was infected with HIV, and it's an R5 virus, meaning it uses CCR5 to infect the target cell. This will be important later as we look at a little bit about CCR5. So from this we know that somehow the presence of dopamine during the initial infection with HIV increases the level of replication out multiple days. To further characterize this effect, we looked at infection with different concentrations and different strains of HIV. So in the upper left, so all of these, the red bar again is infection with HIV alone. The gray bars are infection with HIV in the presence of dopamine. The concentrations of dopamine here vary, and I'll talk about that in the next slide. But notably, what we're looking at in the upper left is infection with HIV-ADA. This is an R5 virus derived from the blood. In the upper right is infection with HIV-YU2, that same R5 virus I discussed, but that's derived from the brain. On the bottom left is infection with HIV-BAL, which is a virus derived from bronchial alveolar lavage, or from the lung. And the bottom right is a virus derived, is SF162, which is another virus derived from the brain. What this is suggesting is the strain of virus has no effect, or no bearing on the effect of dopamine. In addition, the concentration of virus, in the upper left, it's 1 nanogram per mil, 2 nanograms per mil, 5 nanograms per mil, 0.2 nanograms per mil. I have done this up to 50 nanograms per mil of virus, which is quite a lot. There's no effect on the effect of dopamine. So the effect of dopamine happens irrespective of the strain or concentration. In addition, and this was really interesting to us, the overall effect of dopamine occurs despite the donor. As I said, there's a lot of donor variability, But if you look at the effect of dopamine, and this was quite a lot of dopamine I used here, but I've repeated this with less dopamine. If you look at the effect of dopamine in multiple donors, and this is a graph that has pooled data from 8 donors, you see there's a significant increase, about 2 to 2.5 fold, in the level of infection in cells infected in the presence of dopamine versus cells infected with HIV alone. So, how is this happening? There's really 2 possibilities if you think about the logistics of the effect. It has to occur when the cells are exposed to the dopamine. So either when the cells are exposed to dopamine, something happens that causes each individual cell that is infected to produce more virus, or the dopamine causes more cells to be infected, and therefore there's more virus to detect, right? In order to look at this, what we did is we infected cells in the presence and absence of dopamine, and then on days 3, 4, 5, and 6 post-infection, we fixed the cells and stained them for HIV to see how many cells were infected. And what we found, which is displayed graphically in the upper right, is that in cultures that were infected in the presence of dopamine, more cells are infected on days 4, 5, and 6 post-infection in cultures where dopamine was present during the initial infection. This is shown in the middle. On the left, well, in the left column, you see cultures that were infected with HIV alone, and in the central column, you see cultures infected with HIV in the presence of dopamine. The colors DAPI is for cell nuclei. FITC, which is the green color, stains the HIV P24 protein, and Texas red, which is the red color, stains the actin to show the outline of the cell. If you look at the central column and compare it to the left-hand column, what you will see is there is more green in more cells in the center column, which suggests more cells are infected with HIV. Interestingly, as I told you before, we've seen the effects happen with multiple concentrations of dopamine, but it is not dose-dependent. You would think that if you added more dopamine, there would be more of an effect. And unfortunately for us, this is not the case. My life would be much easier if this were the case. It would make a lot more sense. I would have a lot fewer fights with pharmacologists about this effect. There is, and I'll show you later, kind of a threshold effect at which it no longer happens. But once you cross the threshold, the effect of dopamine happens irrespective of the concentration. So, to answer the first question, the effect of dopamine on HIV replication in macrophages. Dopamine increases viral replication by increasing the number of macrophages which are infected with HIV. So how does it do this? When I started these studies, it wasn't clear that dopamine receptors were present on macrophages. So originally what I thought was that it was just reactive oxygen species. I thought that dopamine formed dopamine quinones by reacting with different molecules in the media, and that perturbed the macrophages and caused this effect. That is not the case. First of all, we spent some time looking, and macrophages express all of the dopamine receptors. So in the upper left, you can see Western blots for dopamine receptors 2 and dopamine receptor 1. In the bottom, you can see dopamine blots for dopamine receptors 3 and 4. We also found that macrophages express dopamine transporter, which is in the Western blot on the upper left, and also in the fluorescent microscopy in the lower left. And they express tyrosine hydroxylase. Over in the right, that's qPCR, and then a Western blot for tyrosine hydroxylase in multiple macrophages. They also express monoamine oxidase and COMT, suggesting they can break down dopamine and form the metabolites. I do not know the extent to which all of these are functioning. The extent to the function of the dopaminergic system in macrophages is still something that is not clear, and something that a lot of people are looking at. I am one of them. But it is clear that the macrophages have the tools to have an active dopaminergic system. The dopamine receptors on macrophages are active. In this graph, what you're looking at is the fold increase, or the change in phosphorylation of ERK1 in response to treatment either with dopamine or quinprol, which is a specific agonist for dopamine receptor 2. So going through the Western from left to right, on the left-hand side, you can see the control. That's this. So this is the control condition. If you treat with sulfuride, which is an antagonist for D2, you get a slight reduction that is not significant. If you treat with dopamine, you get a significant increase in ERK phosphorylation, which you can block by blocking the D2 receptor. If you treat with quinprol, which is a specific agonist for D2, you can also see the same increase in dopamine, which you can also block by adding sulfuride, which suggests that the action of dopamine on ERK phosphorylation is specifically mediated through D2, suggesting the dopamine receptor is functional in these cells. So once we knew the dopamine receptors are functional, we thought, great, there's two kinds of dopamine receptors. We'll just figure out which one is mediating the effect, and we'll go from there because it's all defined. But unfortunately for us, the effect is mediated by both types of dopamine receptors, and that's very unusual, and I'll get to that a little bit later. This has also caused me a lot of trouble. It would have been easier if it was just D1 or D2. Originally, we thought it was D2, but D1 works as well. In the upper left, you can see a graph infecting with different concentrations of SKF38393, which is a specific agonist for D1. This shows an increase in the P24 GAG. On the upper right, you can see a similar graph, but the infections are performed in the presence of quinprol, which is a specific agonist for D2-like dopamine receptors. D4 is a little bit different from the other D2-like dopamine receptors, so we performed the studies with a specific agonist of D4, ABT724, and that also significantly increased P24 replication, which suggests that all of the dopamine receptors could mediate the effect of dopamine on HIV infection, which, again, was confusing. Notably, we looked at other monoamines. We looked at serotonin and isoproterenol, which acts on beta-adrenergic receptors, and found these monoamines do not have an effect on HIV replication, suggesting the effect is specific to dopamine. All right, so how does dopamine mediate its effects on replication? It activates macrophage dopamine receptors. All right, so let's get back to the virology a little bit, since this is actually where I started and what I thought I would spend most of my time on. So after we knew that the effect was happening, we wanted to know what stage of the viral replication cycle was affected by dopamine. The HIV replication cycle can be roughly broken up into 3 stages, right? When the virus, so I'll go through it here. When the virus, when the virus binds to the cell initially, it binds to CD4 and CCR5 or CXCR4, and then it fuses with the membrane and releases the viral capsid into the cell. The capsid then uncoats and releases the viral genetic material, which is reverse transcribed from RNA to DNA, then brings it into the nucleus and is integrated into the host genome. After it's integrated into the host genome, there's transcription. There's several rounds of transcription where the RNA is incompletely transcribed. Some of the viral proteins come out. They return and help a full transcription of the virus. The virus is then transcribed and released, translated into viral proteins, and assembled. The virus actually in macrophages is not assembled at the surface. It's assembled in intracellular vacuoles in macrophages, so it's a little bit different. This is a T-cell diagram. But essentially, after it's assembled in the vacuoles fuse with the plasma membrane and release the virus. And then after the virus is released, it finally matures to the action of proteases in the virus itself. So basically, if you want, we wanted to look at different stages of the replication cycle. So we started with entry, which we defined as coming up to uncoating, then the integration step, and then the assembly and maturation step. We started by looking at entry, because if entry was increased, it was going to increase everything after it. The way that we did this is we generated a chimeric virus by combining 2 viruses. One is just standard HIV, so we used HIV-BAL, which is the virus derived from alveolar macrophages. And we also used a fusion protein of VPR, which is one of the viral accessory proteins, with beta-lactamase. What this produced is viruses that have an active beta-lactamase enzyme within the virus. So when the virus infects the cell, after the encoding process, which is the end of the entry process, the beta-lactamase is released. In order to measure infection, what we do is we infect the cells for a certain amount of time. We use 2.5 hours. And then wash the virus away and treat the cells with a molecule called CCF2. CCF2 is 2 fluorophores connected by a lactam ring, so like this. And usually when they're connected, there's a FRET interaction. So this absorbs light, it emits, causes this to absorb the light, and then this emits a color. So as you can see here, When the fret is intact, we get green fluorescence. However, if the cell is infected with beta-lactamase, the beta-lactamase can cleave the lactam ring. This disrupts the fret interaction, and so infected cells turn blue. What you're seeing up here is an uninfected field of cells. And down here is a field of cells where the cells are infected. It makes it very easy to count the percentage of cells that are infected. And so we did that. And what we found is that dopamine increases HIV entry. In the upper left is a field of uninfected cells, and the upper right is a field of cells infected in the presence of HIV alone. These are all cells from the same donor infected with the same amount of virus. In the bottom right or bottom left, you can see a culture of cells infected with HIV in the presence of 100 nanomolar dopamine. And in the bottom right, you can see cells infected with HIV in the presence of 1 micromolar dopamine. And what you see here is that there's a significantly larger number of blue cells in the cultures that were infected with HIV, suggesting an increased amount of entry. So we've done this now. This is a little bit older. I think we've done it about 55 times now. But when we did it 42 times, what we found is that dopamine significantly increases HIV entry. Remember I told you that it's unclear how different people respond to different concentrations of dopamine. So here you only see one concentration of dopamine. What we did here is we took the concentration at which the person had the maximal response and combined them. And so that's what you're looking at here. So again, red is infected with HIV alone, and gray is infected with HIV in the presence of dopamine. And there's a significant increase from it goes from about 12% to about 18%. But if you remember, I talked about different susceptibilities to HIV infection. If you look at this, and this is only very rough, but if you look at this, if you divide it right here, right, into two groups, you can see there's a group of people who were initially not particularly susceptible to HIV infection, the lower group, and then a group of people for whom their cells were much more susceptible to HIV infection. If you break it out and look at those groups specifically, you see that the bottom group, dopamine has a much more significant effect, increasing replication approximately 2.4-fold in those people. It also increases entry significantly in the upper group, but only about 1.2-fold, and that's because there's only a certain amount of infection the cells are going to have in the culture. And if the cells are already highly infected to start with, there's less of a change that dopamine can make. So here's the pharmacology behind it. If you break this out and look at the different concentrations of dopamine, what you can see is at 10 to the minus 8th molar dopamine, so about 10 nanomolar dopamine, which is about the basal amount of dopamine present in the synapses of rats, right? You'll see that at that level and higher, dopamine significantly increases HIV entry into macrophages. Lower than that, there's no significant effect. We know the effect is specific to dopamine because if you block dopamine receptors, all of them, if you block only one type, you still get the effect, which was very confusing for a while. But if you block all of them using an antagonist called flupentacol, which is a pan-dopamine receptor antagonist, you then block the effect of dopamine, and that's shown graphically on the right. So the bars that are solid gray are the different concentrations of dopamine ranging from 10 to the minus 9th down to 10 to the minus 6th. And the bars next to them in hashed gray, so here, is the same concentration of dopamine with pretreatment of flupentacol. And what you're seeing is 10 to the minus 9th has no real effect on HIV entry. 10 to the minus 8th, we see a significant increase with dopamine, but when we pre-treat with flupentacol, we lose that effect. 10 to the minus 7th and 10 to the minus 6th, we see the same effect. And this is a representative slide of that here. You can look up here and see that there's a lot more blue here than in the culture infected in the presence of flupentacol. So the answer to what stages of viral replication cycle are affected by dopamine is that dopamine-mediated activation of dopamine receptors increases HIV entry into macrophages. So this is the exciting part. How does this happen? And this is why I spend a lot more time looking at dopamine than I thought I would initially. So the first thing that we thought was that perhaps dopamine initiates some other type of entry pathway, that HIV normally infects through CCR5, but maybe dopamine causes internalization of the viral particles in a different way. And so what we did is we blocked CCR5 to see if we would still see entry in the presence of dopamine, but we don't. So what you're looking at here is the red is infection with HIV only. This hatched red, where you don't really see anything, is infection with HIV in the presence of a CCR5 specific antagonist, TAC779, that basically completely blocks infection. It reduces it 97%. Again, in the presence of dopamine here, you can see the increase that we expect. And when we add TAC779 to our cultures that are treated with dopamine, it completely blocks it, suggesting that whatever dopamine is doing is mediated by the same entry pathway that normal HIV infection is mediated by. So the next thing we thought is perhaps dopamine changes the expression of CCR5 on the surface of the cells. If dopamine increased the expression of CCR5, it might make it more accessible to HIV and make it easier to get into the cell. So we did flow cytometry analysis of the macrophages and found that at 10 to the minus 8th, 10 to the minus 7th, and 10 to the minus 6th molar dopamine, there's no significant increase in the amount of CCR5 on the surface, which was somewhat frustrating. So then we said, well, maybe the effect of dopamine goes through both types of receptors, but the entry effect is unique to a particular type of receptor. So we tried the entry assay with dopamine and then a D1 receptor, SKF3A393, and a D2 receptor control. And we saw that there was no difference. In fact, the effects weren't even additive. When you use control and SKF3A393 together, you have the same effect, which was, again, surprising. So how is it possible that they can both do the same thing? D1-like receptors, classically, D1-like receptors activate G-alpha-S, D2-like receptors activate G-alpha-I, we would expect opposite effects on cyclic AMP. But when we look at cyclic AMP in response to dopamine in macrophages, there's no cyclic AMP mediated by dopamine treatment. So it's possible that maybe there's more D2, and so maybe the inhibitory effect of dopamine is stronger. So what we decided is we would look at G-alpha-I or G-alpha-S specifically. And interestingly, there's no increase in cyclic AMP in response to SKF3A393, the G-alpha-S connected D1 agonist. We know that G-alpha-S is functional because when we add isoproterenol, which is a beta-adrenergic receptor, which also binds to G-alpha-S, we get the increase in cyclic AMP, which is what you see in the blue bars here. But SKF, the green bars, from 10 to the minus 9th to 10 to the minus 5th, we don't see any increase in cyclic AMP. The orange is forcilin, which is our positive control. So this is really odd. So we look at alternative signaling pathways. They provide a possible explanation, and a number of studies have discovered alternative signaling pathways or mechanisms that may be mediated by both types of dopamine receptors. Calcium signaling, beta-arrestin signaling. There are some people that look at signaling from internalized dopamine receptors, long-term CAMP signaling. That's not us because we don't get any CAMP response, and that kinase signaling. So what do we know about dopamine receptor in macrophages about the dopamine signaling? What do we know about this, right? So this makes sense. We know that PKA is not activated by dopamine in these cells. If it were, we would expect to see cyclic AMP, so we were happy to see this. What we also know is PKC seems to be. And so we see a significant increase in PKC at 1 minute, which is going at 2.5 minutes, and then we don't have enough N at 5 minutes and 10 minutes to see the significance, but it's trending toward that way. And so it suggests that this may be a PKC-mediated mechanism. So the other thing that we know is that, so this might take a minute to explain, we know that dopamine can potentiate calcium release that is mediated through PLC. So what this experiment did is we took HEC cells and we transfected them with either D1-like or D2-like dopamine receptors. Then what we did is we added acetylcholine to stimulate initial calcium release. 80 seconds after treatment with acetylcholine, we added dopamine. And what you see in the parent cells is that when there's no dopamine receptor, there was no effect on calcium. But in the D1 and the D2 cells, when you add dopamine, you potentiate the calcium release, which is what's shown in this blue bar here. In the presence of U73122, which is a PLC-specific antagonist, you block this effect, suggesting it's mediated by PLC. You could do the same experiment with dopamine receptor-specific agonists. So if you take the D1 cells and treat them with a D1 agonist, you get the same potentiation. If you take the D2 cells and treat them with a D2 agonist, you get the same potentiation. So this suggests that dopamine is potentiating calcium flux. So what we know is, going through the literature, you can see a bunch of different pathways, but these are pathways that are suggested to be mediated through calcium and PKC through both D1 and D2-led dopamine receptors. We know there's an increase in PKC. We know that dopamine can potentially increase calcium. So it seems like these might be part of the mechanism. The reason it could be calcium is because, as I said, there's a number of different ways that calcium can be released. D1-like receptors can act on calcium through pKa, but we know that's not happening because we don't see an increase in pKa. D2-like receptors can act through PLC and calcium, and that could be possible, but we need something that acts on both dopamine receptors. Both dopamine receptors can act through G-beta gamma. G-beta gamma is released from the G-alpha subunit when G-alpha signaling happens. Right? And so we think this could happen. It's also possible there's dopamine receptor heteromerization, so D1 and D2 could be heterodimerizing and then signaling in that way. There are some studies that suggest that a D1, D2 heterodimer signals through G-alpha Q, but those studies are questionable because there are other studies that prove that heterodimer is non-existent. So I'm not sure if that can be happening, but it's a possibility. The other thing to note about calcium flux is that there is a little bit of literature on calcium flux in HIV infections, specifically with CCR5. It's been shown that if you bind GP120, which is the HIV envelope protein, to CCR5, you can increase calcium flux. And it's been shown that calcium release mediated in this way is active through PIK2, and the PIK2 activation is PTX independent, means it's not mediated through G-alpha I. Finally, in a very engineered system, in astrocytes that express the dopamine HIV receptors, we found that viral fusion, which is essentially what we're studying, requires calcium release through a G-alpha Q mediated pathway that requires PLC, PKC, and PIK2. And if you block calcium mobilization, you block the viral fusion. So we repeated this study in our primary macrophages using dantrolene to block calcium flux. And what we found is, again, the plain red is infection with HIV alone. This is set to 1. We're looking at full change in HIV infection relative to the infection with HIV alone here. Infection in the presence of dantrolene, which blocks calcium flux, is in the hashed red. We see there's no significant difference from the control. The dopamine in gray causes a significant increase that we're used to, but when we add dantrolene, we block back to baseline levels, suggesting that calcium flux may be something that we're looking at. So, this is what I think is happening. The proof I have is what you've seen. Putting it all together, this is what I think. I think that HIV binds to CD4 and CCR5 and releases G-alpha Q, which binds to PLC, causing IP3-mediated calcium flux, which results in HIV entry. This whole thing down here, the calcium flux that is HIV entry, there's a lot of research that doesn't show anything about this, but it's something that people are very interested in, and we know that calcium flux does increase entry, but we don't know how. What I think is happening with dopamine is that when HIV infection is happening and dopamine is present, dopamine releases G-beta gamma, which binds to PLC, which potentiates the IP3-mediated calcium flux, leading to an increased amount of calcium, which increases HIV entry. So that's my hypothesis. So I don't have an answer to this question, but it may increase entry by potentiating calcium mobilization through G-beta gamma signaling. So in summary, dopamine increases HIV replication in primary human macrophages. It does so irrespective of the donor, the viral strain, or the dopamine concentration. It increases the number of infected cells, and the increase is specifically mediated by both types of dopamine receptors. The increased replication is due, at least in part, because I haven't checked whether or not it's also increasing integration or assembly. It's due in part to a dopamine-mediated increase in HIV entry, and it's a threshold effect. It's specific to the dopamine receptors. It's mediated, again, both by D1 and D2, and it may involve calcium release. So, in the future directions for this project, the things we'd like to do is look at other stages of entry that may be affected by dopamine. So I have studies ongoing that are breaking down HIV entry into different stages. So you can, again, break down HIV entry into the attachment of the virus to the surface of the cell, the fusion of the virus with the plasma membrane, and then the encoding process, and you can examine each of these individually. And if we can identify which stage of the viral entry cycle is involved in dopamine, that's going to give us, we don't really know how dopamine is involved with HIV infection, but there are pathways there that we don't know about. And if we can identify them, those are good therapeutic potential for potentially doing something to interfere with HIV itself and also the effect of dopamine on HIV. I also want to look at the signaling pathways and host proteins that are altered by dopamine receptor activation. This is something that's become more interesting to me over time. I didn't actually expect that I'm not a pharmacologist. I didn't ever look at G proteins before I started this project. And I didn't think I would be looking at them because it wasn't something that occurred to me. But now I'm fascinated by where G-alpha S is when I activate D1 on macrophages and whether or not G-beta gamma is involved and which pathway that it mediates is involved. And those are things that I'd like to look at in addition to looking at dopamine receptor oligomerization. And finally, the last question, which is really the most important one, can targeting the dopaminergic system help HIV-infected individuals with increased dopamine? I'm currently working on an animal model of dopamine neuropathogenesis where I can address this question more realistically. It's a macaque model with my former mentor at the University of Nebraska. The reason this is important, a lot of people ask, okay, if someone gets infected with HIV in the presence of dopamine, they have increased infection. But if you block the presence of dopamine, they're still infected. So why does it matter, right? And it's a really good question. And there are two answers. One gets back to what I told you earlier about reservoirs. Reservoirs are larger, and the progression of HIV is worse if the initial amount of infection is higher. So if you get infected with HIV by using a dirty needle while you're shooting up cocaine or methamphetamine, but you're taking a drug that prevents the effect of dopamine, your progress in HIV is going to be better. Secondly, it could unlock novel pathways and novel therapeutics that we don't understand, ways that the dopaminergic system or potentially other systems are interacting with viral infection, right? One of the ways that we would do this is in America, and I think probably here too, are you guys familiar with PrEP? PrEP drugs are pre-exposure prophylaxis. They're drugs that are given to people in high-risk populations, people who are likely to infect HIV. In America, it's mostly drug abusers and gay men. Right? And these people take PrEP drugs, and the drugs drastically reduce the chance of contracting HIV should you be exposed to it while you're on the drug. If you were able to make a PrEP drug that included some kind of antagonist for the dopaminergic system to block the effect of dopamine, that drug would have an added benefit, and that's something that we're looking into. Overall, we can conclude that changes in CNS dopamine in HIV-infected individuals can significantly impact HIV neuropathogenesis. This is actually important clinically because there are a lot of drugs that you use to treat depression or other psychiatric disorders that interfere with the dopaminergic system. You probably want people treating HIV to know that they should not prescribe those particular drugs. There are other drugs that you can prescribe that probably won't be as bad, right? We know that this occurs through increased HIV replication. I didn't have a chance to show you today, but there's increased production of inflammatory and neurotoxic factors in macrophages in response to dopamine as well. We know that increased CNS dopamine may be a common mechanism by which different types of drugs of abuse modulate the development on hand. One thing that's interesting to a lot of the pharmacologists who work on this is that both heroin, which acts on the opioid system, and cocaine, which acts as a psychostimulant on the dopaminergic system, can increase and accelerate the development of HIV. And it's not clear why these drugs that have significantly different mechanisms of action can both function this way, but they both increase dopamine, and this could be a common factor. Also, it's important to note, and this last one is kind of a broader conclusion, the effects of neurotransmitters on the CNS immune system are really important to understanding neuropathogenesis. It's something that we don't spend a lot of time looking at, but we really should, because as people age, and as we develop more and more psychiatric drugs and treatments, I don't know how many people in Italy are on Prozac, but in America, it's a lot, right? We have no idea how Prozac affects the immune system, but we should, because there are a lot of people on it. Okay. That's it. I want to acknowledge my lab. I have some students, Emily and Midori. Gal is an undergraduate, and Caitlin is my technician, and they helped with all of this. I want to acknowledge all of my collaborators, specifically Dr. Joan Berman, in whose lab I did a lot of the research, and Dr. Howard Fox, who's working with me on the CAC model of neuroAIDS. I want to acknowledge my funding from the National Institutes of Drug Abuse and other funding I get from Drexel University, and I'll take your questions.